Alright guys, so this is Professor Aaron Camp coming to you live from study hall slash my garage. So here we're going to be going over the schematics that we've all drawn together in class. I've uploaded them to Moodle. We've seen them a bunch. So what I'm going to do different here though is try and connect all the dots so you can really see how all of these things go together. Now, all this information, what goes into these different steps of all of these processes that we've discussed in relation to cellular respiration and photosynthesis, mitosis and meiosis, all the inputs and outputs, they're in the PowerPoints, they're also in the lecture guides, but these schematics really show how the process occurs overall. So we're going to review them, and I'll bring your attention to any particular things that I think you need to know for the upcoming exam. So pay particular attention when I mention something being a key factor or playing a key role in any of these different processes that we're talking about. So first you can see I've tried my best to fit the photosynthetic reactions into one file, which is quite tricky because the images are quite large. But what we're looking at here are the light-dependent and light-independent reactions. Now, I know a lot of people have gotten confused because there's lots of different names. In biology, for some reason, biologists try to come up with as many names as they can for each process that they're discussing. So, for example, the light-independent reactions, they're independent of light. You don't need light, so they're also called the dark reactions. You're making sugar, so they're also called the synthesis reactions. And historically, because of figures who have discovered these things, it's also called the Calvin cycle. So here we're looking at our light-dependent reactions right here, right? Now, the key here is that we need light to do this process, right? We have the sun playing a key role. It's emitting photons, but all you need to remember is that it's emitting energy, and that causes things to heat up, and when things heat up, they get excited. So first, just as a reference, where in the world are we? So this little disc right here, this little circle, is known as a thylakoid disc. So this is inside of a chloroplast, the photosynthetic organelle inside of plants, which is inside of a plant cell, which is inside of a leaf. Now, the biggest key here is that these discs, these thylakoid discs, are full of these things called photosystems. Now we have PS2 and PS1, even though PS2 comes first, as you probably remember me mentioning in class that PS2 was discovered second historically, that's hence its name. But the PS2 is also called the water-splitting photosystem because we're splitting water in that photosystem. And the PS1 is just called the NADPH-producing photosystem because that's what we're making in that photosystem. Now, before you start dozing off, because there's so much terminology going on in here, hear me out. So, we have these thylakoid discs inside of the chloroplast, inside of the plant cell, inside of the leaf, and these photosystems are embedded or filled up with these pigments. Now, these pigments are what make plants different colors. Most of the year, it makes plants green because they have chlorophyll in the highest concentration. So these little dots right here, and this little word that says pigments right here, that can be chlorophyll, it can also be carotenes, and it can also be anthocyanins. Now, if you remember from class, carotenes make plants orange and sometimes yellow in color, and anthocyanins make plants red. So ding, 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 super important to pay attention to the fact that this is where we find all of our pigments, embedded in these photosystems in the thylakoid disc, in the chloroplast, in the leaf. So, some other key things that we discussed about these pigments is that carotenes is something we all happen to be familiar with. So that pigment that makes some vegetables and even the leaves in the fall orange in color once the chlorophyll dies down in concentration because the sun changes its rotation and distance from the earth and the sunlight changes in wavelength and so forth, the carotenes are something we're really familiar with. Carotenes we find in vegetables, and a lot of those vegetables are orange in color. Now, beta-carotene is the most popular example that we're all familiar with because beta-carotene is also known as vitamin A. So, Super key, super important, vitamin A. A lot of people think when they see something orange, automatically vitamin C, but that's not necessarily the case. Vitamin C is also orange in color, but it's also known as ascorbic acid. Here we're talking about photosynthetic pigments. So here we have our photosystems in our thylakoid discs that are embedded with chlorophyll, or they're embedded with carotenes. The most popular one we know, beta-carotene or vitamin A, or they have anthocyanins. Sometimes they have all three in those photosystems. So the sun comes down and it actually heats these pigments up inside of these photosystems. When it heats them up, these pigments are just long, complex molecules that are filled with electrons. So their electrons start getting excited and start bouncing around like a pinball machine. Eventually they bounce around from one pigment to the next, 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 and eventually they hit the most important pigment, which is chlorophyll A. Now chlorophyll A just grabs a hold of that super excited electron that's full of energy, passes it off to a protein that happens to be called a primary electron acceptor. Now that primary electron acceptor does just that. It grabs a hold of that electron, and then it's going to shove it through this electron transport chain, which is all that's pictured here. So you have these photosystems embedded in the thylakoid disc, but you also have a couple electron transport chains. And we'll see this in more detail and closer up when we look at electron transport chains in cellular respiration. So for now we have this electron bounced around from pigments, bounced eventually to the most important pigment, chlorophyll A, then went to the primary electron acceptor, a protein that grabs a hold of it and throws it to the electron transport chain. Now this electron transport chain is full of proteins too. In class we do triangle, square, heart protein, right? They just have long, complicated names. But for this class, all you have to do is remember these proteins grab a hold of this electron. That electron shoves its way through those proteins, and that causes these proteins to pump these hydrogen ions that are floating around inside of the plant cell into the thylakoid disc. Now that increases the concentration of hydrogen ions. But before we continue there, let's stop for one moment, because we need to remember that water is being split to constantly supply these electrons. So these pigments have electrons inside of them, right? These negative charged particles that are just inside of those molecules anyways. However, when the sun heats the pigments up and then they donate those electrons to the next stage in the light-dependent reaction, you need to replace them. And we replace them by watering our plants. So when we water our plants, right here, this is where this water plays a key role. Water functions to replace the electrons in the photosystems. Now as a byproduct of that, we end up with oxygen, right? So oxygen goes out of that plant sometimes, and we breathe it in. In addition to that, that oxygen also fuels cellular respiration in plants, because remember, they also have mitochondria, and they also do cellular respiration. It's a couple different things plants can do. Talk about that when we get to the end of the light-independent reaction. So, now we have this electron shoving its way through those proteins in the electron transport chain. Originally came from these pigments that were supplied with new electrons from splitting water in half. Now, when we shoved our way through these proteins in the electron transport chain, those proteins work like vacuums. They start sucking in hydrogen ions from elsewhere in the plant. When we suck in enough hydrogen ions, you remember from class that we have a really high concentration of hydrogen ions. And in concentration gradients, we know that things in biology want to move from high concentration to low concentration. So those hydrogen ions are going to leave that disk that they're sitting inside. Now when they leave that disk, they do so through this enzyme called ATP synthase. Now this ATP synthase enzyme right here just works like a rotor. So hydrogen ions are its battery. They push their way through this rotor. It causes it to rotate. Every time it rotates, it grabs a hold of a phosphate group, shoves it onto this ADP molecule, and then makes an AT molecule, which is what we use for energy inside of our cells, right? So hydrogens are shoving their way through here, working the ATP synthase rotor, which is rotating, grabbing a hold of these phosphate groups, shoving them onto ATP, okay? Now, we're not done with this electron. It came from the splitting of water, went into these pigments, came through the proteins in the electron transport chain. Now it's going to jump to the next photosystem, the NADPH producing photosystem, or PS1. Now it does the same thing it did in the last photosystem. It bounces around from pigment to pigment, from pigment to pigment, and then it eventually bounces to another electron transport chain. This one functions a little differently, and all it does is take this free NADP+, which is just some fancy confusing sounding molecule that means electron carrier molecule, and it shoves hydrogens on the end of it, because we're going to use NADPH and ATP in the light independent reactions. So we have these electrons bouncing around the photosystems, jumping through the first electron transport chain, making some ATP, going to the next photosystem, bouncing around those pigments, which could be chlorophyll or carotenes like beta carotene or vitamin A, or anthocyanins, and then going through this last electron transport chain, which also rotates, but the way it rotates, it just grabs a hold of these NADP+ molecules and shoves hydrogens on the end of them. Alright? Now, a key thing that you may have not put together at this point is the fact that we're going to use this ATP that we made in the light-dependent reactions over here in the light-independent reactions and over here in the light-independent reactions. In addition to that, we're also going to use this NADPH molecule, this electron carrier molecule, to also help us rearrange molecules in the light-independent reactions. Okay? So the light-independent reactions, the dark reactions, the Calvin cycle, the synthesis reactions, all the same thing. Just involves us taking CO2. Where do we get this CO2? Every time I ask everyone, they always go, It's super important to remember here that you want to remember your common sense. I know when you're looking at these really complicated, drawn-out processes that sometimes you can lose sight of the grand scope of these things. So a couple super grand scopes, or important things to remember of practicality of these different steps, and probably the reason they're an integral part of general biology, is that we get water right here that we use to fuel plants, right? And it's integral to this whole process. In addition to that, we have CO2 playing a large role in the light-independent reactions. And where does that CO2 come from? Us breathing out. It's as simple as that. So, humans, animals breathe out CO2. Then what happens is this enzyme comes by. It's called ribisco. It happens to need water to work, right? So ribisco enzyme needs water to work, kind of like the battery for ribisco. Is going to shove the carbons from this CO2 molecule into this already existing molecule inside of the plant cells. Now, where did this molecule originally come from? It came from the plant when it was a seedling. It just happens to be passed on from plant to plant. So this molecule, this RUBP, is a 5-carbon molecule that happens to have 2 phosphate groups on the end of it. Now this thing's always rotating around in this circle. Now remember, this is all done in this thylakoid disc thing right here, right? But this is occurring outside, floating around in the stroma, right? In that little liquidy substance inside of the chloroplast. Now, the plant grabs a hold of these carbon molecules from the CO2 and shoves them into this molecule right here known as RUBP, ribulose biphosphate. All you need to remember is RUBP, 5-carbon molecule. You shove 1 carbon into a 5-carbon molecule, you make a 6-carbon molecule. Now, you make a 6-carbon molecule, the little squiggly lines indicate that a 6-carbon molecule with 2 phosphate groups on the end is unstable. So it goes ahead and breaks down. It breaks down into 2 3-carbon molecules. It just splits in half. Simple as that. So when it splits in half, you make these 2 3-carbon molecules called PGA. Now these things go off and do other cellular processes inside of the plant, but we do not use them to make glucose, which is the most important part of photosynthesis, right? So CO2 comes in, shoves its way with this enzyme into this molecule, becomes a 6-carbon molecule, makes these PGA molecules go off to do other things. Now, because we can't use PGA to make glucose, we use these products, this ATP, from the light-dependent reaction and the NADPH from the light dependent reaction as well, and we use those particles, those compounds, to rearrange those short carbon chain molecules. So we rearrange PGA to make this stuff called G3P. Now G3P is important because when you combine two of those together and rip off a couple phosphates, then you make glucose. Super simple and straightforward when you look at the fact that two 3-carbon molecules together make one 6-carbon molecule, make glucose. Now an important feature of this process is that every time you rip off a carbon, you only use one carbon, right? So if you were asked how many carbons it takes, or how many CO2 molecules it takes to make one molecule of G3P, then you would say 3, because it takes 1, 2, 3 molecules of carbon to make one molecule of G3P, 6 CO2s to make one whole molecule of glucose, because you need to regenerate this 5-carbon molecule every time you start this process over and over and over. Okay? So, some other key parts of this process are the differences in photosynthesis. Now, we discussed the fact that what we're looking at here is pretty much C3 photosynthesis. But in C4 photosynthesis, like corn and other agricultural plants, what we do is we take in the CO2 in one part of the plant and we turn it into sugar in another part of the plant. That is, it's separated spatially. However, in CAM photosynthesis, like cacti and other desert plants, we take in CO2 at night and we make sugar in the daytime. So those different steps are separated temporarily or at different points in time. And that's a key adaptation for C4 and CAM photosynthetic plants because it allows them to minimize water loss, and it allows them to basically do different steps of the light independent reactions the way they need to that best suits them or adapts them to that particular environment. So that's the overview of photosynthesis and kind of combines all of those ideas into one. Okay? All right.